here we are with Noel Neff, student ambassador.
Yeah.
How has it been going?
It's been great. It's a great experience, and meeting everyone. All the other ambassadors have been super nice. All the professionals as well been very nice, taking questions and stuff like that too. So it's been great.
Have you had a chance to present your poster yet, or is that today?
That's today, so I'll do it later today. So this is technically my first time presenting my poster.
Okay, well, why don't you give me a dry run, Noel? Tell me about the can neurodegenerative disease be revealed through NGS for forensic DNA typing? Tell me about that.
Okay, so this project kind of developed. Do you want me to hold, or do you want?
I'll hold it for you.
Okay.
You can talk with your hands, or.
This project kind of developed in a class I was taking at Towson, where we looked at the different NGS methods, so next generation sequencing, and we specifically looked at the MiSeq FGX, which is pictured. It's not, but this is the data. Okay. So, but I didn't have a picture of it. But okay. Which is used for forensics with Verogen's Forensic Signature Prep Kit, and we were looking at different diseases. Can we detect anything? Because recent research has been coming out about how forensic markers such as STRs and SNPs, they're originally in these non-coding regions of the genome, and it's believed they were chosen for forensics because they shouldn't contain any like additional information about a person, such as like phenotypic information or medical information. But research has been coming out showing that they possibly can be associated with some of this information. Okay. So we were kind of exploring that topic, and my project further developed. I originally just had two types of diseases here. So we have frontotemporal dementia and dementia with Lewy bodies. And then I kind of developed and just picked a bunch of other neurodegenerative diseases such as Parkinson's, ALS, and Alzheimer's. And then kind of just explored specific SNPs we researched within the Forensic Signature Prep Kit, Verogen's kit. We found about these 5 SNPs, and the genes that they're within have been linked. You can see familial neurodegenerative diseases. So we kind of just thought to explore that. Yeah. And then, and we found some other, this was just like the data saying that other research is being done on these markers. And this is basically my data. So we found within these 5 SNPs, there was, and these are, I compared them to healthy samples. Okay. So these are various healthy samples. Some of them had other like conditions, such as like bipolar, but overall they didn't have a neurodegenerative disease. Okay. So we're using them as healthy samples. And basically, like my conclusions, I guess, most of them had similar calls. As you can see, all the non-highlighted ones are similar calls. But then these AAs were interesting because like for this first SNP, none of these healthy ones had AA calls. And whereas like same with this SNP, AA calls, but then you can see only one of them did. So like we'd have to get like a bunch more samples. That's the only thing with this research. It definitely needs more research to be done on the topic to get a bunch more samples. But overall, that's about it. We also looked at NGS as like an upcoming thing that everyone's been doing, kind of compared it to like capillary electrophoresis. I did CE on all the samples as well, and got similar, like, or the same STR calls. They're just not pictured here, but I did do that data. And then over here was just like the intensity or the number of reads for each sample. You can see like this one I ran twice, and something was just wrong with that sample.
Sure, that happens. Real science, right?
These calls, that weren't in we didn't get any SNP calls. Okay. But yeah, overall, NGS can be used to obtain really valuable information compared to CE, because you get that whole sequence and all these SNPs that you don't get with CE. And it's just really interesting because more research needs to be done on topics like these with all the other research that's been coming out, linking phenotypic and medical information to these forensic markers.
That is fantastic. Thank you very much for that explanation. Yeah. Now I have a couple questions. Yeah. These SNPs were chosen because
so we did some background information. They were within the kit we were using, so the Forensic Signature Prep Kit, and then we did some background information, which is like this chart, and just found like, some of these have been linked to or the gene has been linked to neurodegenerative diseases. So it's part of the reason we went with that.
Right. Yeah. So I guess in that whole genome, there's probably other SNPs that so many that we have no idea yet. Right. Yeah. So that is important to be looking through this data. Right. Now I'm questioning this one right here. Could this possibly be a case where they may actually have, a neurodegenerative disease that they're not aware of yet?
Yeah, I kind of said that in my conclusions. That is like a really hard point to make, though, obviously.
Yeah. Yes.
I don't want to diagnose someone with a neurodegenerative disease, and I'm not doing that.
Right.
But there's considerations that have to be made, like, this person, whatever age they were, I have that in my overall data set, but whatever age they were at the time this was taken, the sample was taken, they may not have had symptoms of this disease, but could possibly develop it. So maybe that's why that AA is seen. But again, really, I'm not making that call. You just have to consider things like that.
I totally understand that because, things like Parkinson's or other things, they don't show up until later in life as well, right?
Yeah, later on.
Yep. So you may want to have this information earlier in life, and maybe treatment for Alzheimer's or any of these diseases then, can preemptively help with those situations. So. Yeah, this is fantastic. How many hours do you think you were in the lab doing this kind of stuff?
I don't know.
Well, you're still taking classes, right?
I actually, yes, I've been working. Most of my poster and data was during this semester before ISHE. Okay. But then I did all of the wet lab work over the summer. So I was in the lab over the summer doing all of that. So it was really fun. But yeah, it definitely took a long time. My program requires 160 hours of research, so somewhere around there.
Wow, that's impressive. So where do you find yourself in the future? Noelle, do you want to continue down the path of discovery, or are you thinking about joining some sort of forensics lab? What are you looking for in the future?
I definitely love research and love doing that, so I have considered maybe furthering that and getting a PhD and going to the research field, or working for a company like Verogen or Qiagen or Promega, any of those amazing companies, and doing research and testing with their kits coming out. But right now I think I want to be a DNA analyst to start off, so I'd like to go within that field right now.
Yep. Get in the lab and do the actual analysis. Okay. Help solve some crimes.
Yeah, hopefully.
That's a common theme around here, solving crimes, right? Or identifying people.
Right.
Well, thank you so much, Noelle. It's great to have met you and get to know a little bit about your research here, and I hope you'll continue on your path to discovery. Thank you so much, Noelle.
Thanks. Thank you.